All right, we are moving into our very last unit. And so we're going to be talking about some biochemical molecules. Now, there's more than what I'm going to be talking about, though some of the things that I'm going to be talking about does really apply to some of the ones that you'll see today. So there's many, many other molecules. Now, we're going to be talking about some basic structures and a little bit of some of the functions that they may have, but truly that is to go more in depth with knowing more about structure and function. That is something that's covered in biochemistry, structure and function with Dr. Shelby. And if you're a B&B major or a chemistry major, you're required to take that course. But outside of that, there are a few students who like to take that course, and you'll learn more about that. So let's kind of go into a couple things. So, like I said, congratulations. You're only a little bit away from finishing up the semester. And so we have this week and we have next week left to go. We'll have some more presentations. But I want to thank you again for being a good group. I had said that once in an email, but I want to say it again, just because you, for the most part, most of you have been doing the things you need to do, listening to me. For those of you who are still kind of struggling, still have time to come and see me, although we're running very, very much out of time. So remember, this is still your lecture, meaning this video is your lecture. So you should be taking notes, because in class we are going to be doing practice problems. Because remember, the point of flipping your classroom is that the easy part, which is your lecture, is done. You know, the metacognitively easy part is done at home or in your room or wherever. And the cognitively hard, which is doing problems, we do in class and practice sessions and so on, so that you're not left with the hardest part to do all by yourself in the beginning. Now, eventually you do need to practice problems after we've done some practice problems as well. And so please note, though, that in this unit we'll be kind of touching on previous content as well. So this is a good review for some of the topics that we've seen before. So what is biochemistry? Now, that's a loaded question, because depending on who you talk to, there might be a different answer. But I'd like to really just say biochemistry has some crossover between bio and chemistry, which all chemistry does and all biology does. Well, most biology do. But this is truly where how things happening on the small scale affect a lot of behaviors in the large scale. And so for us, we can kind of think of it as this broad topic that overlaps chemistry and some biology topics together, which can encompass cellular biology, chemical biology, genetics, molecular biology, pharmaceutical chemistry. Unlike what we do with you guys here at Florida Southern and almost every other school in this country, we like to silo our classes, but realistically, a lot of our classes and topics really merge over when you're in grad school or when you're working, that they really merge between one topic to the other. So oftentimes there's not really a clear distinction. So oftentimes when people say, I'm going to graduate school for biochemistry, it can range from bioorganic all the way to truly like cellular biology. It just depends. And so there's a huge overlap. And this is why biology students, as long as you're more in the molecular track of things, need to have some organic chemistry, because of these molecules that we're going to talk about and understanding truly how they behave. So we're going to focus only really on two and a little bit on the third one, just to kind of give you a feel for that. But you have other molecules like nucleotides and things like that that are part of this category. We're just not going to get into that in this semester.
What is kind of the basic of life? And I'm not talking about from a philosophical perspective. I'm talking about from a chemical perspective. It's basically small gases came together and eventually made biochemical materials or biochemical machinery that then took these small molecules as well and made other molecules. So an example of that is we started utilizing biochemical transformations, enzymes, and other organic molecules to produce additional organic molecules, and one of them is using photosynthesis, which is a process using all sorts of enzymes and other organic materials to produce a molecule that we need for energy. And so this particular reaction also produces oxygen gas, which is very useful for life. And so what are these types of molecules that are produced this way? These are carbohydrates, and carbohydrates we oftentimes like to call sugars, but carbohydrates is a generic term for high energy biomolecules, and they oftentimes will add rigidity to molecules. We also see them um, modified a little bit, like in crustaceans, that we've replaced some of the groups on them. And we also see them being used in a variety of different medical applications or pharmaceutical applications. So, for instance, there's some drugs that are packaged inside carbohydrates for drug delivery. We have some drugs that are attached to carbohydrates to make it easier to get to the part of the body that you want. Another place that we see them is that it's basically the backbone of RNA and DNA, which is ribose. And so in DNA, it's deoxyribose, and RNA, it's just ribose. Now, what is a carbohydrate? So the generic term carbohydrate is a collection of different molecules. It is some simple molecules that we call monosaccharides. And then we have some more complex molecules. Those are going to be disaccharides to polysaccharides. So eventually these monosaccharides can act as units or monomers for the synthesis of polysaccharides, which are the polymers of these carbohydrates. And so the monosaccharides are essentially ketones or aldehydes with a lot of OH groups on there, so we call them polyhydroxy ketones or aldehydes. So that's the simplest term. So when they form polymers, these are actually occurring through a condensation reaction, which we've seen before. So essentially to make ethers, and that is some of the things that we'll talk about in our videos. One of the reasons this is also important for us to understand these is not just from biochemistry, but a lot of green chemistry is starting to use more benign products. And so the synthesis of carbohydrates or use of carbohydrates, we're starting to see emerge in a variety of different places. So utilizing these things rather than polystyrene to potentially make other molecules for plastic utensils and things like that. So let's look at a monosaccharide. So what is a monosaccharide? It's the simplest form. Like I said, it's a polyhydroxy ketone or aldehyde. So this is the aldehyde in glucose. And in fructose, it is a ketone. We end the name of the generic names for these things in O's. When you see O's, it represents ketones or aldehydes in this polyhydroxy thing for these monosaccharides. It's most common. So if it's an aldehyde, it's called aldose. When it's a ketone, it's called a ketose. When it has 5 carbons, it's called pentoses. And when it has 6, it's a hexose. So for instance, on glucose, it's a aldohexose because it has 6 carbons and it's an aldehyde. Stereochemistry is very important on these, and we can see, we've seen these Fischer projections before, that we can have the same type of structure, but just orient the OH groups in different orientations, and you'll get different molecules. So some of these are enantiomers of each other, and some are diastereomers. So for instance, this molecule here compared to this one, we've changed one of the positions. So this is a diastereomer. The enantiomer of this molecule, which is have all the OHs on this side. And so you can see that they're different ones. So the enantiomer of ribose would be L-ribose, and this L&D is a little bit different than when we saw L&D before. So this capital L and capital D are just representing the configuration of my sugars. D sugars are the ones that our bodies primarily use, and it's that last chiral center having all the OHs, if you draw them in this format, to the right-hand side. We call that the D-ribose. When they're on the left-hand side, they're the L-ribose. But it's not the same thing as the small d and little l, which were representing of rotation of light. This is the orientation of the last OH group. In the previous video, we looked at monosaccharides, and we talked about before these acyl nucleophilic substitution reactions that can happen on these polyhydroxycarbonyls when we were in Unit 2, and we're going to talk about that again. So one thing that we see with these monosaccharides is in the presence of water, and it's accelerated in acid as well, is that we get these intramolecular reactions in which we get these rings. And we get these rings as a consequence of our OH group acting as a nucleophile, attacking our carbonyl and opening it up, and then we get a proton transfer. And so these carbohydrates can be written in the Fischer projection, they can be written wedge and dash, then they can also be converted to a closed structure. And these happen interchangeably, mostly in acid, so we can get this opening and closing in acid. But it happens a little bit in water. Now, when the alcohol attacks the carbonyl, you open the ring, and this OH was originally our carbonyl oxygen. And this carbon that it's attached to is called an anomeric carbon. This anomeric carbon is going to give us different stereochemistries depending on which face the OH attacked. And so remember, the carbonyl is planar, so the OH can attack from the top or the bottom, so that new OH that we produced at the anomeric carbon can either be pointing up or pointing down. So this is what that relates to. So we have what's called the alpha form and the beta form. The alpha form is when the OH points down on the anomeric carbon, as long as you're drawing it in this format where you have the ether on the top. And that ether was originally our OH. When my OH is pointing up, that's the beta form. And so again, it depends on which side it is formed on. Now these can happen interchangeably because the ring can open and close and open and close. And because they can open and close, we call this process mutarotation, and it works better in acid or base to catalyze. Now, some sugars are primarily going to be in the alpha form normally. Some sugars are primarily going to be in the beta form. There's not a good way to determine that for you. So for instance, glucose oftentimes we find in the alpha form, and then sometimes we have like a ratio of them. So we can draw our structure a couple of different ways. So just to review the kind of formats, we can draw it in the Fischer projection, we can draw it in the bond line, or we can draw it in the Haworth. And the Haworth projection basically takes the molecule and is assuming that this part of the molecule is kind of coming towards you. It's not showing you too much of the stereochemistry of the ring, which the bond line will do, but it shows you directions of the OHs. And so when you draw these, there is a kind of a simple trick that you can use to draw this. Take this molecule, essentially turn it on its side. So take this molecule, turn it on its side, and then you're going to draw all the groups back. So I'm going to put the OH back in, and then the other OHs. This other OH that we had here, this is the one that's going to act as our nucleophile, and then this is that little tail that points upwards off of this structure. And so the way that we draw this is if this OH comes and attacks and closes the ring up, if the OH groups are already on the top when you draw them, they're going to stay on the top. If they're on the bottom, they're going to stay on the bottom. So this is 2, 3, and 4.